Hello and welcome to the show. Today, we're diving deep into the past, but perhaps not in the way you're thinking. We often hear about ancient DNA, or aDNA, and how it's unlocked secrets about human evolution and extinct creatures like woolly mammoths. It's truly revolutionary. However, DNA is surprisingly fragile. It breaks down relatively quickly, especially in warm, wet conditions. This means vast stretches of biological history and countless fossils found in less than ideal environments remain genetic mysteries. But what if there was another way? Enter the exciting field of Paleo-Proteomics. This science focuses on recovering and studying ancient proteins preserved in fossils, sediments, and even old artifacts. Proteins, the workhorse molecules in our bodies, are generally much tougher than DNA. Think of collagen, the protein that makes up much of our bones and connective tissue. It's incredibly durable and can survive for millions of years under the right conditions, vastly outlasting DNA. So why is studying these ancient proteins so important? Well, firstly, they give us direct biological information when DNA is simply gone. We can finally investigate fossils that are hundreds of thousands or even millions of years old. For instance, scientists analyzed proteins from a 1.77 million-year-old extinct rhinoceros tooth found in Georgia. This protein data helped figure out its evolutionary relationships, something impossible with DNA from that era. Similarly, proteins from 1.9 million-year-old fossils of the giant ape Gigantopithecus blacki placed it firmly on the evolutionary tree near orangutans. Secondly, proteins tell a different story than DNA. DNA holds the genetic blueprint, but proteins show what the organism was actually doing or making. They can reveal details about physiology, diet, or even immune responses. Imagine analyzing fossilized dental plaque. Proteins found there can identify milk, indicating nursing, or specific plant and animal remains, revealing what an ancient creature ate. How do scientists actually do this? The key technology is mass spectrometry. They carefully extract any remaining proteins from samples like bone or teeth. These proteins are often broken down into smaller fragments called peptides. A mass spectrometer then measures the precise mass of these peptides, allowing researchers to figure out their amino acid sequences. By comparing these sequences to known protein databases, they can identify the original protein and the species it belonged to. And it's not just proteins. Researchers are also looking at other ancient molecules like lipids, fats, and oils. Lipids found in ancient pottery can tell us about cooking methods or what people stored in those pots thousands of years ago. Now, paleoproteomics isn't without its challenges. Contamination from modern microbes is a huge hurdle, requiring incredibly clean lab work. Proteins do still degrade over time, making recovery tricky for very old samples. And interpreting the complex data requires powerful computers and large reference databases. Despite these hurdles, the study of ancient biomolecules beyond DNA is rapidly growing. Technology is getting better, allowing us to detect fainter traces and analyze them more accurately. By combining information from ancient proteins, lipids, and DNA when available, we get a much richer, more detailed picture of life in the deep past. It allows us to explore evolution, extinction, ancient climates, and the intricate details of life long gone, truly reconstructing history beyond the genome. That's all for today. Thanks for tuning in.